Hi, my name is Jacob. I'm part of the Straw Lab here at UCLA, and we've been studying development using model organisms known as C. elegans, which are just these microscopic worms. And where I've come in the last 8 weeks is I've been looking at the interactions between metabolism and development, specifically in embryos. And what we already know about embryonic development is that they use specific metabolites, so like nutrients, such as glucose, lipids, maybe even acetyl-CoA. And they use these for genome activation, cell differentiation, or even the formation of organs. But what we do not know is, in embryos, how strict is the pre-programming for using maternal nutrients versus how much does the embryo actually dynamically regulate and monitor its own development by understanding its metabolite availability. So based off how much availability of the nutrients, do embryos actually adapt to the development isolated from the mother? Yes.
Low-key, the way you said it was way complex.
Really?
Yeah, I would have been, I think it would be better if you can simplify that part.
Okay.
I think what you're trying to say is, you're trying to find out how, like, much the embryo depends on its mother, right?
Yeah, if it depends on its mother,
or
doesn't
if
have
it
any
can.
independent.
Yeah, I think it's better to say it in a simpler way.
Okay.
Well, I mean, maybe it's just me, maybe because I don't understand.
No, yeah, then if you're not understanding it, who else is going to understand? So what we're trying to figure out is something critical in the field with embryos in particular. Do embryos actively monitor their own metabolic stores that guides development? Or is it all happening passively through the mother's nutrients and information coming in?
That's better.
And so our hypothesis is that embryos have this autonomous, this active role. We do believe that, and we want to test that. And how we're going to test that is by one developmental stage in particular, known as sleep. And sleep in this case is not like mammals, it's not like rats or humans. It's not an 8-hour dormancy. It's like these bouts or these small rhythmic bouts of like local motion. So reduced local motion. You'll be moving for a little bit, twitching, and then stop. Twitching, stop. And that's what we're trying to look at and see if we can manipulate that sleep behavior by manipulating the metabolic nutrient availability. What's going on in the embryo, you know? And so that's what we're doing. We have this little sleep assay which we have here, which records movement. And so we're going to record the movement based on 16 hours from the embryo is laid as an egg to when it hatches. And we see this yellow like patch right here, and that is actually looking at sleep. And that's all it's showing is small rhythmic twitches, and we can manipulate that by looking at this pathway. This is a biochemical pathway in larval stages of C. elegans. Again, we're looking at embryos, so this is already seen in previous literature with larval stages. We want to see if this pathway, which is responsible for looking at different conditions of nutrients such as starvation, when something is in starvation, such as a larval stage, it's going to go through this pathway, activate AMPK, FOXO, AKT, these genes, to allow for that sleep behavior to go through. Now, if we can verify that this pathway is also in embryos, as we've seen it in larval stages, this will be a direct hit on our hypothesis saying that embryos do have this active role. They can sense nutrients via this pathway, and they could change the developmental progression, isolated from the mother. And so that's what we did. We took these sleep assays, and we took our wild type embryo. This is the baseline sleep signature that we got. And we actually saw differences in sleep throughout mutating these 3 genes, which are very important for this pathway. So they became dysfunctional in these assays, and we saw different results in sleep, showing that sleep phenotype actually changes when you manipulate the metabolic nutrient sensing ability. And that's very important, and that was our first try at looking at our hypothesis being true. There were some things that was little we didn't expect. We can get into that if you want to ask about that. But more importantly, we wanted to verify this in a second fashion by doing another cross. So we, instead of mutating 1 gene at a time in these, we mutated 2 genes in 1 embryo. And that would give us a better verification of if this pathway is available. The thing is, we don't have enough time, and so I wasn't able to get the sleep assay for this. But if we can show what we've seen in previous literature, showing that there's no sleep when you knock out both AMPK and FOXO, if we see that in the embryo as we've seen in larval stages, then we're good to go. And that will show us that our hypothesis is on the mark. But in the meantime, my favorite part of this experiment these last 8 weeks is actually imaging the neuron, which is responsible for sleep, and it's called the RIS neuron, and it's converged on this pathway. This pathway is that which depolarizes the neuron and allows sleep to happen. And we actually image that using GCAM, which is binding calcium signals that are fluxing through the neuron when it's active, and we can actually see the neuron. So this is a snapshot, and using GFP, that green fluorescent protein which binds to calcium and then fluoresces green when you shine light on it, we actually see the soma, the body, the axon, and the dendrites. And again, this is just a snapshot, so there's a 6-minute video of this if you want to use a QR code and like take a look at it. It's not going to only show you that the neuron itself is like twitching and like when it's firing, the embryo stays still, which is the slip. You know, you fire, stay still, and like it twitches because the firing is like kind of like rhythmic. So you can actually see that. And we also did a sleep assay for this because we want this sleep signature to actually look like a wild type, because this could be used and served as a baseline imaging for further processes. We can cross this GCAM strain with the triple mutant. We can cross this GCAM strain with the AKT mutant and see what's the different phenotypic readout of neural activity. And so that would be really, really cool and interesting to see. And so that's part of our future directions. I think if we're looking in terms of humans and how this relates, if we can understand that this infers that humans are also, human embryos are also able to actively monitor the development without the mother, this allows us to better understand how to intervene with different conditions when a mother is starving. You know, when a mother maybe has hyperglycemia and taking in too much nutrients. What is the developmental constraints on that? And that allows us to go further into like epigenetic mutations, neurodegenerative disorders, and find the understanding how to intervene in pregnancies when development goes astray. And I think, thank you. I do think that this type of research is very important for what's going around the world today. We are talking about starvation in development. There are pregnant women around the world who are starving, in Palestine in particular, who are not able to get the food that they need for their embryos to grow into a world with a quality of life that they can deserve, that they can have. But we're not able to have that right now, and I think that this speaks openly about that. And I guess I'll clean that up, but that's what I got right now.
Yeah, it was a very good presentation.